Hi everyone, my name is Spencer. I'm from Dr. Giacca's lab. The topic of our study is cell-specific role of insulin in an in vivo model of restenosis. Patients with insulin resistance, type 1 or type 2 diabetes are shown to have greater risk of getting post-angioplasty restenosis. Our previous in vivo studies have shown the vascular protective effects of insulin, mainly in the endothelial cells mediated by PI3K-AKT pathway. On the other hand, the mitogenic effects of insulin are mainly manifested in vascular smooth muscle cells and are shown to mediate by the MAPK pathway of insulin. The objective of this study is to investigate the in vivo endothelial and smooth muscle cell specific effects of insulin on new intimal growth in a murine model of restenosis. Our insulin receptor fluxed mice were crossed with mice overexpressing tamoxifen-inducible Cre recombinase under the control of type 2 Cre or smooth muscle Cre promoters. 3 days prior to the femoral artery wire injury surgeries, the mice would be implanted with either insulin pellets or blank pellets. 28 days after the surgery, perfusion would be done to collect the femoral arteries for histology analysis. As we can see from the graph, insulin significantly decreased the new intimal area in iron flux control mice, while these beneficial effects of insulin are abolished in mice with endothelial-specific iron deficiency. Unexpectedly, insulin receptor deficiency in smooth muscle cells also prevents the vascular protective effects of insulin, suggesting beneficial effects of insulin in vascular smooth muscle cells appear to prevail over the mitogenic effects, at least in insulin-sensitive conditions. Alpha smooth muscle actin is a differentiation marker of smooth muscle cells. As we can see from the graph, positive area of alpha smooth muscle actin was increased by insulin in all groups, except in iron flux smooth muscle Cre positive mice, indicating that in vivo insulin has a differentiating effect on vascular smooth muscle cells. In conclusion, our results suggest insulin may be beneficial in endothelial cells and vascular smooth muscle cells in insulin-sensitive patients with diabetes undergoing percutaneous intervention. Thank you for paying attention.